Welcome back to the Classics of Immunology Journal Club. And now we're, as you well know, we're in real time. And this paper that we're going to do today came out in Science on the 25th of November in 2022. Before we get started, don't forget to go on the YouTube channel and like the video if you like it. Click on the bell, get notified for more. Subscribe to the channel. That makes the YouTube people happy. Then scroll down and check out my website and look at the writing tab on my website, and you'll find all the classic papers of immunology and plus the more recent ones that we're doing now. So this paper today is by Claudia Arevalo and 16 others, and then the senior author, the leader of the lab, is Scott Hensley. And this comes from the University of Pennsylvania in Philadelphia. And the title of this paper is a multivalent nucleoside-modified mRNA vaccine against all known influenza virus subtypes. As I said, it came out in Science in November 25th, 2022. This is the paper I've been waiting for since the beginning of the pandemic of coronavirus, because it seemed to me that what we really need to do is we need a so-called universal vaccine that will deal with all the strains of whatever virus we're dealing with, and in addition to new mutant strains that might not be present at the time that you're making the vaccine, like what happened with Omicron, for example, in coronavirus. So the background on this paper is that the influenza virus, if you don't know anything about it, or the only thing you really know is that you're supposed to get a booster shot every year. Now, and the question is, why is that? It's a little bit strange. And the history of the influenza vaccines are very interesting in that they were first developed in the 1940s during World War II. They were developed because the government was scared to death that they were going to have a repeat of the 1918 flu epidemic in 1941-42. And so what they did was that they made a flu vaccine from flu virus. They needed to make a flu vaccine, but they didn't nobody really knew what the flu virus was at that particular point in time. They had some assays for it that were developed by Sir Macfarlane Burnet from Australia, because he developed an assay for the virus. The virus, turns out, has on its surface little spikes, just like the coronavirus has, that binds onto red blood cells. And so the virus will agglutinate red blood cells. And so Burnet developed the assays for that and so forth. And they found then that they could grow up this virus in chicken eggs, and fertilized chicken eggs, so that they were little embryos inside the shell. They would take the chicken egg at a certain time after the fertilization process, and then inject straight through the shell into the embryo their viral preparation that could grow up lots and lots of virus. And what they did was that even though Pasteur said you needed to have a live virus in order to be able to make a real vaccine, and they didn't want to do that. They didn't know how. They didn't have time to attenuate a live virus the way Pasteur said that it had to be done. So they fixed the virus with formaldehyde. And then they did clinical trials in the GIs, the recruits coming into the boot camps. And it turned out that Jonas Salk, and I've told this story before on this journal club, Jonas Salk was a young lieutenant who had been drafted into the army and he was then assigned to do the clinical trials, some of the first clinical trials in GIs that had signed up to be sent over to Europe and Japan or in the Pacific to be cannon fodder. Turns out that the vaccine worked. And so we didn't have a repeat of the 1918 epidemic. The problem is that the flu virus is a unique virus in that its genome is not just one long strand of DNA or RNA, as in most other viruses. There's 8 discrete little genes that are packaged into a viral particle, and they each code for a different aspect of the virus. And so the importance of that is that if you have a cell that's infected by one strain of virus, and then another one comes along and you have 2 strains of virus in the same cell, you can get mixing and matching. So the viruses essentially have sex inside this cell. and they exchange their mini chromosomes. And so that's why you're going to have new viruses developed that can lead to a pandemic, as it did in 1918. And it's known that there are at least 20 different variants, subtypes of influenza virus based on the antigen on the cell surface, on the surface of the virus that binds to the red blood cells, the hemagglutination antigen, and it also binds to cells and facilitates infection. In addition, there's another antigen or protein on the surface of the virus called neuraminidase, and that cleaves off sialic acid off of sugars that are on the presence of cells, allowing the virus then to become incorporated into the cellular membrane and then internalized, and that's infection. So it's HA and N, and there are a bunch of different variants of those, the HA antigens and the N antigens, and the question is which ones are out there circulating in the community. And so what they do is they go out into the field and they take samples from all over the world in the summer and fall, and they come back into the laboratory and decide which strains are the most prevalent. And that's what they make the new vaccines are out of, that then they give to you beginning in October, November, December, before the flu season really hits, when people are all crammed together inside and they're breathing in viral particles, just like the coronavirus. So the problem with that is that you have to get a new shot every year, and sometimes and often they don't have a real good mix of the viruses that are out there. You might get infected with one of the viruses, flu viruses that is not in the vaccine that year, and then you can get flu. Not such a good thing. So this group from the University of Pennsylvania decided to see if they could make a so-called universal vaccine, and in the introduction of this paper, they say that that there are people that are out there trying to make a universal flu vaccine by taking gene segments that are conserved in all the viral strains and using them to immunize with, which is a good idea because those conserved sequences are conserved and they are not mutated in nature because they're required for the life cycle of the virus, for the replication of the virus and so forth. And so that's one way to try to make a universal vaccine. And the other way, what these people have done in this particular paper is that they've decided to make multivalent vaccines. So they take all 20 of the strains and they what they've done is they've made messenger RNA vaccines by the hemagglutination, the HA antigen from all 20 of these different strains. And they basically say in the intro that this is feasible because of the mRNA technology that we didn't really have before, because before, most of the time, there would just be a few of the most highly conserved sequences or the most common sequences. In the materials and methods of this paper, they make mRNA vaccines out of each of the HA variants or subtypes, and they're for each of the A strains, the B strains that are known to be out there. And what they did was they then individually immunized mice with each strain and then looked to see if they made neutralizing antibodies on the one hand, and also whether or not they were susceptible to infection by whatever HA strain it was. And they found that, and they used a low dose because rather than use the normal dose that had already been established for other viruses and vaccines of around 50 micrograms, they just used 3 micrograms per each one because they wanted to see whether or not if they reduced the dose, they could still get decent immunity out of these mice. And they did, and that worked. So then they went ahead and vaccinated all vaccinated with a vaccine with all 20 messenger RNAs in the vaccine simultaneously. Each of the individuals' mRNAs were only 2.5 micrograms combined times 20. That meant that the total vaccine would have 50 micrograms. that would they use then to immunize mice. Let's go to screen sharing now and show you some of the results in this paper. So this is figure 1. The title of this figure shows or says that the 20 HA mRNA vaccine elicits long-lived antibody responses that react to all 20 HAs. And so in the A panel here, you have antibody levels that have been taken from mice vaccinated with the 20 strain vaccine, and then the mice have been bled after 1 month, 28 days, and then tested against each of the different strains. You can see H1, H2, H5, H6, and so forth. And down here, H3, and then a B, 2B strains. And you can see that there are really good antibody levels against all of the HA mutants or variants. And then if you look down at and compare these vaccine results to when the vaccine was only against the H1, they get good antibodies levels to H1, but not a great deal of cross-reactivity to the rest of them. And then here's they immunized with H3, and they found that H3, they got good antibody levels in this group 2. And then in D, the vaccines were from the B subtypes or strains. And then if they were just injected with PBS as a negative control, they got no antibody levels. And here they are at 118 days or 4 months after immunization with the 20 HA vaccine. And you can see that they have really good antibody levels throughout and against all of the different subtypes and strains. So that's really the essence, one of the essences of this paper. And then they wanted to find out, I'm going to skip figure 2 and go on to figure 3. They wanted to find out whether or not those antibody levels that were really done by ELISA, so they're just binding antibody levels, whether or not they protected the mice against a lethal challenge of a matched HA strain of virus versus a mismatched in the HA subtype. So here's what in figure 3, the animals were immunized, and then they were challenged with the matched strain. And here we're looking at body weight. And this is a standard way you do flu vaccine experiments over the years. This has evolved. And you can see and the days post-challenge, the negative control in the green triangles, the mice just continuously lose weight from almost day 1. Whereas with the 20 HA mRNA vaccine, there is a little dip. This is in a purple, a little dip here, but then they come right back and they do really well. If you then score the mice in terms of giving them a clinical score, you can see that the H1 vaccine and the 20 HA vaccine were just about the same in terms of they didn't really get very sick, whereas the animals that got a mismatched HA or and here's a B, and this is luciferase as a negative control. They all got really sick. And you can see here, then in the survival of the mice, the only ones that really survived were the ones that got an H1 vaccine versus the whole 20 HA vaccine. And then over here, same kinds of experiments, but this time the challenge was with a different virus, a different H1 virus. And you can see that the immunization did pretty well against in all of these different mice in terms of the 20 HA vaccine. Down here, they lost weight, but then they recovered. And that's the important thing. And here's the H1 mRNA, some cross-reactivity. So the clinical score was similar to the matched viral challenge. And then The survival data are here. So that's it. And I'm going to stop the screen share at this stage. The bottom line on this, in terms of the discussion in this paper, there's more data in this paper. Number one, you should read it if you're interested, because of all the data are very, very convincing, and that's why it's in Science rather than JI or some, one of those second-rate journals. So, in the discussion, they make the following points. They say, this is an alternative way to try to make a universal flu virus vaccine, rather than just taking several conserved epitopes or conserved antigens to try to make a vaccine that will protect against all of the strains of a particular virus. They go on to say that, so you make it to all the known strains, but what happens if there's a new strain that arises, like CoV-2, that arose, which is similar to CoV-1 but different. And they said that probably what will happen if you use this so-called universal vaccine is that you won't get necessarily what they called sterilizing immunity, but the vaccinee will be protected against severe disease. And that's important. And I'll get into that in a little bit as we go on. They said that they've done these experiments, and they don't know how many different epitopes, antigens that they can put into a vaccine, and they need to do more experiments. It's like the Cookie Monster, more experiments. They also they say that the immunological mechanisms that are responsible for the protection by these vaccines, they need to do more experiments to really elucidate that whole thing. They did depletion experiments taking out CD4 cells and CD8 cells, and it didn't seem to make a whole lot of difference. But they were very careful not to say that that their results are all due to neutralizing antibodies, and I don't think they are. So the conclusions is that you can use the new mRNA platforms to deliver conserved genes as well as variant genes, and activate both T cells as well as B cells. And it's important to really study the T cells as we've gone on. We just did a journal club on Dan Baruch's experiments, because T cell immunity is what clears the virus. And eventually, if you become infected, you get better and you become well again, because the T cells essentially deal with the infected cells and kill off the infected cells and allow you to recover from the infection. And therefore, you get prevented from having severe disease if you've got a lot of T cells on board that are recognizing multiple epitopes of the virus, in particular conserved epitopes. So I think you can combine both of these ideas, doing the conserved epitopes on the one hand, and the as many different epitopes as you possibly can get into the virus or into the vaccine, and have it still functional. And so I'm sure they're working on these kinds of things, because you don't have to be a rocket scientist to figure all this out. And that we can expect in the future that maybe with flu, wouldn't it be nice we didn't have to get a booster every year? And with COVID-2 or the new maybe COVID-3, would be protected from not necessarily infection, but severe disease. And as I mentioned before, it's the disease that really is the important thing in terms of vaccine efficacy, because that's how we've tested all of the vaccines that have been produced over the years, is by showing that we don't get the disease in the populations that's been vaccinated. So I'll stop there, and don't forget to like the video, click on the bell, subscribe, check out the website, and we'll see you next week. If you're new to immunology, or you just want to delve into a certain aspect of this large field, and you want to get a grasp on things, you should buy my new book, Molecular Immunity, because it's a chronology of all of the important experiments and discoveries that have been made in the last 60 years. It's concise, but it's also very comprehensive. And it's the only such book that's been written since 1970. So you can buy it on Amazon, and together with the YouTube videos of the Classics in Immunology Journal Club, you can't go wrong.